Thank you.
All right. Okay. So welcome everybody. I'm really glad that you could be here. And I was before we got started here, I was telling Eddie how that's really great. It's really great that the Yerba Buena chapter of CNPS are doing their speaker series, continuing that. There are a lot of organizations out there that just sort of shut down and weren't able to continue, but with their programs. And not only that, just last I checked, there are over 200 of you out there, and that's not possible with the CNPS regular meetings. The building's not that big, not to mention parking issues. But anyway, really glad that you could all come to see my talk. So just a little bit more background about myself. So I got my master's degree at San Francisco State in 1999, as Eddie mentioned. And my topic for my master's thesis was fungi associated with Arctostaphylos in Northern California. So most of you probably know that Arctostaphylos is Manzanita, and is in California is the center of diversity for that shrub here throughout the West, actually. So it's found in other places, that genus, but the vast majority of the species and subspecies are found in California. And it is a mycorrhizal, an ectomycorrhizal species of plant. And I'll talk a little bit more about that as we go. I did a flora for that. So essentially I collected as many individual mushrooms as I could find, crawled around on my hands and knees. It was a very Zen experience, I have to say, finding everything I could find for a couple of seasons. And I made 400 collections of about 135 different species. And I tried my best to find pure stands of Arctostaphylos, because a lot of times nearby pine trees or Douglas fir trees or other shrubs and so on could sort of skew the results, and some of the fungi might have been associated with other plants. It could have been if I hadn't gone for pure stands. Anyway, it was a really great experience, and I learned a lot. And there were, at the same time, there were some people who at San Francisco State, some students who were looking into the mycorrhizal complement on Arctostaphylos root tips. So they would make soil core samples, and they would take a look at the root tips and try to figure out what was on the root tips using DNA fingerprinting techniques. And so it turns out that Arctostaphylos, for perhaps uniquely, but probably not, is extremely, has an extreme diverse complement of fungi that are associated with the roots in a mycorrhizal relationship. All right, now before I get started with talking about mycorrhizae, I have a little primer on what is a mushroom, basically. So first of all, most fungi propagate by spores. There are a lot of fungi that propagate vegetatively as well, but most have spores, and that's their way of disseminating the species. A spore, the definition of a spore is that it's a single cell that's capable of mitosis, cell division, and that's typically referred to as the germination stage of a spore when it begins to undergo mitosis. And of course, spores are microscopic. You need a microscope to see that. Oh, by the way, if you see words in red, that will be on the midterm. Okay, so make sure that you keep track of those. All right. Okay, now spores, once they germinate, they form chains of cylindrical cells that we call hyphae. And it turns out that all multicellular fungi, so that excludes the single cell fungi like yeasts, and other organisms. So for example, some water molds and some slime molds and so on, also have hyphae. And again, you need to have a microscope to see those. Now, in this photograph here, it's a light micrograph. You can see that these cylindrical cells in mass are going to create a network of a very high surface area ratio network of these cells that are going to be in contact with the substrate. And so that allows them to absorb nutrients and water and all those sorts of things very, very efficiently, in contrast to, say, cells that are tightly packed together and perhaps cubical or spherical or something like that. So these hyphae are very, very efficient in the substrate at what they do. Now, the hyphae then grow into a mass of tissue called mycelium. And this is the business end of the fungus. This is the part of the fungus that is out in the substrate and feeding, creating enzymes that they release into the substrate. The food products are broken down, and then the fungus absorbs those back into the hyphae of the mycelium. I'm sure most of you have turned over a log in the woods and you've seen something like this. And so that's a classic look at what mycelium are like. And notice how highly branched they are and how at the tips they're very feathery. So those are microscopic hyphae that again have a high surface area that enables them to absorb their food. Now, when the mycelia from two different individual spores come in contact with each other, and assuming that they're mating compatible, they will fuse together. And that's, ladies and gentlemen, mushroom sex. When fungus is then ready to reproduce, it then forms fruiting bodies. Now the next slide, I forgot to talk about fruiting bodies for a minute. So most people, when they use the term mushroom, they're talking about something like we see here in the upper right image. So it's something that has a stem, has a cap. Sometimes people will only call fruiting bodies that have the gills, look like blades on the underside. So think of, store-bought mushroom. Those, is typically what people call mushrooms. But other people will sort of expand that out and talk about other things that don't have those characteristics. So for example, from left to right, we have a coral fungus there. In the middle is a jelly fungus. And then over on the right side, those are puffballs. So those are definitely fruiting bodies, and you can call them mushrooms if you want. Their whole job is basically to produce and to disseminate the propagules, the spores. All right, I'm going to spend a little bit of time on this slide going through the mushroom life cycle. If you already know the mushroom life cycle, or if you don't really care, you can maybe check into the 49ers game, see how that's going, or maybe the election, see how that's going. Anyway, I'll be a little bit of time on this, but if you're interested, follow along with me and we'll go through the life cycle for a typical mushroom. All right, so let's start here with the production of spores. Now, these two spores are color-coded. One's red, one's blue. That's not real. We are not going to see little red spots and little blue spots in spores. And by the way, those spots represent nuclei. But the reason that this is done this way is that these are two mating compatible spores. Okay, so they get spread out into the surrounding substrate. If the conditions are right, so there's enough water and there's enough food and the temperature's right, and who knows what else, we're not really sure, those will germinate. And they'll form the hyphae, the cylindrical cells that I talked about. At some point, if two of these mating compatible clusters of mycelium meet and they're mating compatible, then they'll fuse together. And that's what's referred to as cytoplasmic fusion. Or for those of you who remember your biology, this is plasmogamy. That's the technical term for it. Anyway, basically the two cells will fuse together. Now, I want you to notice something here. Note that the red dot and the blue dot maintain a separation from each other. They do not fuse together. So here's a big difference between, say, a sperm cell and an egg cell, which are mating compatible cells in humans, for example, and other organisms, plants and animals. So when an egg cell and a sperm cell fuse together, they undergo the cytoplasmic fusion. They immediately undergo nuclear fusion at almost right away. So the nucleus of the sperm cell fuses with the nucleus of the egg cell. In fungi, they don't do that, or at least not all the time, always. There are some exceptions to all these things, right? But they form what we refer to as dikaryotic cells. Di means two, karyo means nuclei. So cells with two nuclei. You can shorthand for that is N plus N, right? So this mycelium will grow in the substrate, and it may grow for weeks, days, years. Who knows? Could even be hundreds of years before the conditions are right for it to produce a fruiting body, a mushroom. But if the conditions are right, then that occurs. To kind of give you an example how this might happen is in some mushrooms that are growing in a substrate that has limited resources. So say like on a leaf, right? So that mushroom grows on the leaf, the mycelium are out there and they're consuming the leaf. Sorry, let me back up. There's no mushroom yet. So the mycelium is consuming the leaf. As those resources deplete, that's when the mushroom forms. That's when the fungus forms the fruiting body in order to then produce propagate for the next generation, right? So that's kind of a simplistic view of what might trigger a fruiting, right? So the mushroom is produced here. And if we look on a mushroom, somewhere on there, so for example, if this is, say, a white button mushroom that you, like the one you get in the store, it's going to have gills. If you have a porcini, it's going to have little tubes. A puffball, the spores are going to be inside, and a coral mushroom and a jelly fungus, the spores are going to be on the outside of that. In any case, somewhere on there will be these terminal cells called basidia, again dikaryotic, N plus N. And then at that point is when nuclear fusion happens. So that's when the two nuclei, the red one and the blue one in this case, fuse together and form what's called the diploid stage, okay? Diploid meaning two sets of chromosomes in the nucleus. Before that, all of the nuclei are haploid, which means one set of chromosomes in the nucleus. All right, and almost immediately after the nuclear fusion, or certainly within minutes or hours, it depends on the organism, on the mushroom, meiosis happens. And meiosis is the reverse of nuclear fusion. So it's basically nuclear fission, where what was once one diploid cell nucleus now splits into two and then four haploid nuclei, still on that terminal cell. And then those nuclei in this particular type of fungus migrate out to the tips of these little finger-like projections, form spores, and then are released. And there we go. All right. Okay, for those of you who've been at the football game or the election results, you can come on back. I'm done with this part, or you can wake up if you fell asleep. All right, moving on. So let's talk about mycorrhizae. So the term mycorrhizae refers to a relationship. It's a relationship between a fungus and a plant, typically at the roots, And in fact, that word mycorrhizae, myco means fungus, rhizae refers to root, like same root as rhizome, or yeah, you get the idea, okay? And this is a mutualistic relationship, usually. Remember my title, it's usually mutualistic, but not always. And I'll talk about the exception later on. The mycobiont is the fungal partner in this relationship, and it receives carbohydrates from the photosynthetic partner. Most of the mycorrhizal fungi that are out there are obligate in the relationship. In other words, they cannot do without this relationship. There are some exceptions. There's some that are the opposite of that, which is facultative, which means that they can live without the mycorrhizae for perhaps a while, and then when the opportunity arises, they'll form the mycorrhizal relationship. The phytobiont is the plant partner, receives a more efficient mineral uptake, and in some cases, a more efficient water absorption. And there's also a lot of evidence that that plants who have this relationship have an enhanced disease resistance. There's been a lot of studies looking at, what happens when you don't allow a plant to grow with the fungi. Well, they continue to grow, but they tend to be stunted. They tend to be slow growing and not nearly as healthy as those that have the mycorrhizal relationship. Turns out that about 90% of all plant species have a mycorrhizal relationship with fungi. So really, that's incredibly important in an ecological sense, right? That the vast majority of plants, so when you look out the window, the vast majority of these plants are going to have fungi associated with them at their roots, right? Okay, so let's talk about the nutrient exchange here. So the plant then, again, provides sugars, and I'm going to use the word photosynthates to talk about that because, as we know, the sugars that are produced by plants are the result of the process of photosynthesis. And so that's what the fungus gets, right? And then the fungi are a source of these nutrients, such as nitrogen, phosphorus, and a few others that I'll get into in a minute. It's not really well understood, and it appears that some fungi are better at, say, facilitating nitrogen uptake for the plant, and then other species might be better at facilitating the uptake of phosphorus or potassium or some other nutrient, right? And a certain type of mycorrhizae called Ectomycorrhizae also are very beneficial to plants in terms of water uptake. And I'll talk a little bit more about that later. But just note that there's a structure called a mantle, which is made up of these hyphae, the hyphal cells, the cylindrical cells. Again, a lot of surface area there, and they help to absorb the water that then the plant can take advantage of. All right, I'll talk more about that in a minute. All right, look at that illustration. You might want to go check out the football game again. But if you're interested, I'm going to work my way through this little illustration here and give you an idea of how this all works at the roots. All right, so let's start with, let's look at just sort of a general. So there are 4 places here. So there's in the gray, there's the root tissues. Over here in the blue, these are the fungal cells, the hyphal cells that are involved with this relationship. In between is a small space between the fungal cells and the root cells. We call that the interfacial apoplast. That will not be on the midterm. I would not ask you to memorize that term. Okay, good to go. And then, of course, on the other side of the hyphal cells, we have the soil and with nutrients in solution. All right, so let's start over here with the root tissues. And we'll talk about how the photosynthates basically end up in the fungus. As most of you know, the phloem is the type of vascular tissue that through which the photosynthates are being transported from, say, the leaves to the roots, or from the leaves to the flowers or other growing parts. But the phloem that's in the roots will deliver the photosynthates, in this case sucrose, via the interfacial apoplast to the fungus. In the interfacial apoplast, there is an enzyme that's been secreted by the fungus, I believe. I'm not real sure about that, but I think so, and called invertase. And the invertase converts the sucrose into monosaccharides, glucose and fructose, essentially, right? And that's it in terms of from the plant to the fungus. From what the fungus provides to the plant is a little bit more complicated, but it's the same basic idea. So we have a sequence of nutrients over here. So here is ammonium, here is a nitrate, we have amino acids, which is what proteins are made out of. We have some peptides, which are protein amino acids. K plus is potassium ion, and then P sub I, that's a potassium, a phosphate molecule. All right, so these things are pulled into the hyphal cells. And by the way, these arrows, this is all, almost all passive transport. So it's moving from a high concentration to a low concentration. For the most part, there is some active transport that goes on, but most of it's passive. Okay, so once these nutrients are inside of the hyphal cells, there's some processing that goes on, some metabolizing. So for example, the ammonium and the nitrate are converted into amino acids, and sometimes the vacuole is involved. And then those sometimes processed metabolites, sometimes the raw, like potassium ions and the phosphate molecules, are then moved across the interfacial apoplast and into the root tissues to the xylem, where then in the dissolved water in the xylem, they're transported to the rest of the plant. Now, you may be asking yourself, well, what about the color of the circles here? So it turns out that the blue circles are pathways across cell membranes that are well established and well understood. It has to do with putting the genes in yeast, and then the yeast produce these pathways, and then they put the nutrients in there, and they see that it works, basically. The filled in gray circles, those are highly probable pathways. So there's pathways where there's not much doubt that that's how it works, but no, we're not really sure. And then the circular ones, it's just not known, at least as of when this paper came out. Let me stop there real quickly because I see a lot of activity in the chat, and I was wondering if there are Eddie or Noreen or Bob, you might have some questions for me at this point.
Yeah, we do have a very good question from Alice, and I think it's contextual. So if the question is, from Alice, is there a reason that plant root tissues can't uptake all the nutrients from the soil solution themselves? Perhaps their epidermis is thicker than the hyphae, or they just don't have as much surface area as the hyphae?
Well, there's certainly a surface area issue there. Yeah, so the fungi, the hyphal cells, are going to be a lot more efficient than even root hairs. So if you're familiar with how roots grow, if they don't have these mycorrhizal relationships, they form root hairs out at the tips of the roots, which are pretty small. They're basically cellular extensions, but the hyphae have a much greater reach, if you will, into the soil. So there's certainly that. Beyond that, I'm not really sure. I wish I knew the answer to that question. I think some of it has to do with the processing that fungi can do that the plants cannot do. So, for example, with producing amino acids, specific amino acids. So that can be done here in the hyphal cells, but it cannot be done efficiently anyway in the plant, especially when it gets to the xylem. Once it's in the xylem, it's just transported by the water, and there's no metabolism that happens in there. So any metabolism any converting, any processing that would occur pretty much has to occur before it gets to the xylem. And so I'm pretty sure that some of the processing anyway is going to happen much more efficiently in the fungi. Now, if you look down here, the potassium and the phosphate molecules are pretty much direct from the unchanged from the soil to the into the roots. So perhaps they can be absorbed by the roots. But again, I think you were kind of right on in that the reach of the hyphae is much, much greater.
Yeah, nice question, Alice. Another attendee asked, how is the interfascial apoplast created?
Oh, it's simply the space between them. And, it's kind of like this. There's some space between my fingers, not very much, but there's space. So, so that's all it is. It's just that by the way, the FP means fungal plasma membrane and the RP means root plasma membrane. So there's just a little bit of space between there. It's microscopic space. Yeah.
Great. I think that's it for now.
Okay. All right. So now there's 6 types of mycorrhizae: Arbuscular, Orchid, Arbutoid, Ericoid, Monotropoid, and Ectomycorrhizae. Okay, so let's get through, talk about each of these one at a time. Now, the most abundant type is what's called Arbuscular mycorrhizae, sometimes called AM. It turns out that about 300,000 plant species, which is a lot of plant species, have this relationship. It turns out that about 85% of all plant families have at least some members that have this kind of a relationship. So just to give you an example, all grasses have AM mycorrhizal relationships. Redwood trees have AM mycorrhizal relationship. And there's a lot of other ones, a lot of crop plants and so on that have this kind of a mycorrhizal system. And there aren't very many fungi, though, comparatively. There are only about 130 species of fungi, as far as we know, that are involved with this. So the fungi are generalists for the most part. But these plant species that have this really depend upon them a lot. So the structure is so called because of the arbuscules. And if you look at that word arbuscule, it has the same root as Arbor Day, right? Which is the tree day. So it refers to trees, and I'll be talking about those a little bit more later. Here's a cartoon down here that shows a couple of the arbuscules. Some species also form vesicles. So here's a vesicle right here, but not all species do this. Sometimes in the literature, you will see this this type of mycorrhizal called VAM, as opposed to AM or AM, VAM, which means vesicular arbuscular mycorrhizae. There's always extra matricial hyphae. So extramatricial means that these are hyphae that are between the cells. And so the yellow is the fungus and the blue is the plant. So let's look at the plant first. So this is the epidermis. There's a layer, what's called the hypodermis in some plants. And then there's a big chunk of a root called the cortex. And then inside of that cortex is the mycorrhizal, the vascular tissue. So the fungus, which is in yellow, will extend its hyphae into the cortex, well, sometimes pretty deeply. And for the most part, in between the cells. This kind of, I'm not crazy about this cartoon, but it's the best one I could find. I'm not crazy about where it looks like the hyphae are inside of that cortical cell, but it's not there. The only time that the hyphae actually are inside of the cells are in the arbuscules. So let's talk about the arbuscules for a minute. So these are, they penetrate the cell, cell wall, but they don't penetrate the cell membrane. Okay, so let's think about that for a minute. So all the cells have a cell wall, mostly made out of cellulose. And then just inside of the cell wall is a thin layer of phospholipids called the cell membrane or the plasma membrane. So what happens, and so here's an electron micrograph, and here is a scanning electron micro. This is what's the other type of electron microscope? I'm blanking. Anyway, scanning electron microscope and transmission electron microscope. But you can see that they're highly branched, and in each of these cases, and if you look at my fingers here for a minute, imagine that the plasma membrane of the plant is wrapped around all of these branches of this fungus. That is incredible surface area contact between the fungus and the plant. And so that's just remarkable. And that's inside of the cells. Right? The vesicles, which tend to be outside of or in between cells, look like this. This is another transmission electron microscope. So they're basically swollen hyphae. They can be intracellular, but again, they're not going to penetrate into the plasma membrane. They're mostly in between the cells. They probably are for storage. There's some evidence that they may be involved with asexual reproduction. They may serve as propagules for that, although I'm not sure how that would actually happen, but there's some evidence. All right, and then the extramatricial hyphae. These are between the cells, like I mentioned, mostly. There is some growth outside of the root, but it's not very extensive. It's basically just enough to kind of get all of the nutrients into the root. By the way, you see these. There's a couple marked here with an S. Those dark spots, those are asexually produced spores. It turns out that AM fungi, with very few exceptions, I can only think of one that I know of, they don't produce fruiting bodies. So they don't produce, they don't have the kind of sexual life cycle that I showed you earlier. And they mostly propagate asexually by producing these basically swollen hyphae that act as spores. All right, second type, orchid mycorrhizae. Orchids are so cool, right? I mean, if you've ever done any work with propagating them, that they're a challenge to grow them. One of the problems, one of the constraints with growing orchids is that the seeds have very little nutrition, if any. And most plant seeds have enough nutrition to keep the embryo alive for it to germinate. But orchids don't have that. So they have to have a mycorrhizal relationship in order for them to grow, in order for them to get to the point where they can germinate and then produce their own sugars through photosynthesis. And as many of you know, you maybe have caught this already, there's some orchids, I'll talk about this a little bit later, but there's some orchids that never photosynthesize, and they're always dependent on these fungi for their nutrition, but in a very special way that I'll get to a little bit later. So, so anyway, so the orchids that do have green leaves that can photosynthesize, they have to start their lives off with this relationship with a fungus. It's been known for these fungi to actually eat the seed, but the other thing about orchids that you may know about is that not only do they produce seeds with no nutrition, but they produce a lot of them. And so they produce, somewhere on the order of like a gazillion, maybe even a bazillion seeds, each one of them from their fruits. Actually, I'm kidding. I'm exaggerating. Sorry. Sometimes I do that. But anyway, they produce a lot of seeds, and so there is the capability for, or the they can afford to lose some to this process. One thing I want to point out, if you look at this illustration here, you can see that the hyphae form these dense coils in the cortical cells. And so this is very much like the arbuscules, high surface area. They don't penetrate into the plasma membrane, but they form these coils. And I want to point that out because other mycorrhizal types, such as the ericoid and the arbutoid, also form hyphal coils, much like those ericoid, much like those orchid mycorrhizae. Now, ericoid and arbutoid mycorrhizae are different from each other, and I'll talk about that in a minute, but they, as the name implies, they are found in members of the Ericaceae family. So ericoid in, for example, erica, the heather, and a lot of other types like that. And arbutoid mycorrhizae are found in arbutus, which is madrone, right? And also in Arctostaphylos, which is closely related to madrone. Anyway, they both form hyphal coils. The ericoid mycorrhizae, which are sometimes referred to as endo-ectomycorrhizae because they have characters of both. I'll get to that in a minute. Arbutoid mycorrhizae have a sheathing mantle. Again, I know I'm kind of teasing you with some terms here, but I will talk about what sheathing mantles are a little bit later. And I will also talk about a Hartig neck, which is lacking in arbutoid mycorrhizae. All right. There's some fungi, by the way, that can both produce ericoid and ectomycorrhizae, which again, I'll get to shortly. All right. So here we have on the right, ericoid mycorrhizae. You can make out the blue stained coils. They look like little spots, but trust me, those are coils. And there's no sheathing mantle. Okay, so you don't see a lot of hyphae around the outside of this plant cell. By the way, this is what's called a hair cell, or sorry, a hair root, because you can see there's not very many cells there. The cortex is basically a single layer of cells. Over here is arbutoid mycorrhizae, and again, this is a transmission electron micrograph. You can see the coils pretty well in the cortical cells. And then you can see this pigmented area labeled M here. That's the mantle, the sheathing mantle, right? All right, now remember my the title of my talk, right? Mycorrhizal fungi. I'm not sure I can remember it myself, but anyway, the subtitle is has something to do with the fact that most of relationships are mutualistic. Both partners get benefit. Here's the exception: myco-heterotrophic epiparasites. Oh, and that will be on the oral exam. Mycotrophic epiparasites. Right. So let's break that down. Myco, fungi, heterotrophic. Heterotrophic means that they eat other things as opposed to making their own food. And all fungi are heterotrophic, so it's kind of a silly term. Epiparasites. Epi means on, and you know what parasites are. So these are actually parasitic fungi that form a mycorrhizal relationship. They form what are called monotropoid mycorrhizae. They have both a sheathing mantle and a heartig net. Again, I'll get to that. And they form a single cell penetrating peg. So rather than an arbuscule or even a vesicle, they just send, or a coil, they just send a single peg into the cortical cells, each cortical cell. Now, these are found in monotropoideae. Used to be monotropaceae. You remember that? Well, they're monotropoideae is a subfamily. And many of you are familiar with these. These are the plants that have no chlorophyll. And they cannot photosynthesize as a result of that. They're achlorophyllous. By the way, monocrucade. Do not call them saprophytes. And that was sort of the term that was used for a long time. And it doesn't really make any sense because the root of sapro means dead and phytes is plant. And there's nothing dead about them, gotta say. Anyway, on a crusade. So now this is actually a tripartite relationship. The achlorophyllous plant is actually stealing carbohydrates from a mycorrhizal fungus that is getting their carbohydrates from a different plant. All right, so think about this. So by the way, let me show you this. Now I'll do a little demonstration for you. So here is that single hyphal peg in a cortical cell. This is a root mass from of a Monotropa uniflora, which we don't have here in California, unfortunately. It's the ghost pipe or Indian pipe, it's called, found throughout the eastern part of the continent. Achlorophyllous monotropoid plant. So here's some California species. So here's snow plant. There's that ghost pipe again. And here is pine drops. And there's a bunch of other types of plants. And over here is an achlorophyllous orchid, right? Corallarhiza, or coral root, sometimes called. So all of these plants are basically surviving because they're parasitizing a fungus that is mycorrhizal with a nearby pine tree or a nearby Douglas fir tree or a nearby Arctostaphylos plant or something like that. So pretty crazy, pretty crazy. All right, finally, let's get to Ectomycorrhizae. And this is what most people think of when they think of mycorrhizae, is this category. There are about 2,000 plant species that have this relationship. So just a fraction of what we see with, for example, the arbuscular mycorrhizae. Most of them are trees, but there's also shrubs such as Arctostaphylos as well. But look at how many fungi species are involved with this. So when I sort of talked about my thesis early in the talk, I mentioned to you that Arctostaphylos has a really incredibly high diversity of fungi on the roots. By the way, between the two projects, my project and my colleague, looking at the root tips, we found over 100 different species of mycorrhizal fungi just in our work, just a couple of years of work. It's probably much more than that. Anyway, a lot of our highly sought after edibles, so chanterelles and porcini and several other ones are mycorrhizal. All right, so let's look at how mycorrhizal structure is, how it is structured in the roots. So here are those two terms that I've been teasing you with so far, the heartig net and the sheathing mantle. So the heartig net is best represented here in the sort of orangey yellow areas between the cells here and over here, and to a lesser extent up here. That's the heartig net. If you were to, it's called that because if you were to look at a cross section of a root, say this root right here, under a microscope, the plant cells are white. And if you just sort of blur your eyes and just look at the colored cells, kind of a net-like structure, right? And in 3 dimensions, it's even more so. And then the mantle has to do with the cells that are on the outside of the root tip of the root. So there's a very thick sheath of, here in blue and sort of the purple and so on, is a very thick mass of hyphae that wrap itself tightly around the root tips of the plant. By the way, this illustration is a little bit confusing. The top part and the left side of this one, the top part of the longitudinal section, left side of the cross section is angiosperms. And you can see that the Hartig net only goes into about the first layer of cells. The bottom part of the longitudinal section and the right side of the cross section, that's a gymnosperm, a conifer. So that's what we would see in pine trees. So angiosperm over here, a flowering plant, a conifer over here, right? Okay, so here's a Hartig net. So the hyphae basically surround the plant cell. So think about in 3 dimensions, the plant cell's in the middle, the Hartig net's around it. This is where the nutrient exchange happens. So unlike with arbuscular mycorrhizae, there's no penetration of the hyphae into the cells of the cortex. They wrap around it. You can kind of see it here. C refers to a cortical cell, and then the dark areas are the Hartig net. In conifers, it's throughout the cortex, but in angiosperms, just the outer layer. Oh, there I have a duplicate slide. Sorry about that. Several duplicate slides. Okay, this is where I was trying to get to. All right, so this is talk about the sheathing mantle. And so you can see it here in very dark pigmented area here. You can see the root cortical cells with the Hartig net. And you can see how thick this mantle can be. This right here is on the right side, that is a photograph through a dissecting scope. And so you can see that basically the root tips are swollen in nature. And that's because of these very thick mantle, sheathing mantles from the mycorrhizal fungi. And that increases the surface area and it increases the water uptake for that. And that's why Ectomycorrhizae are really beneficial to their hosts in terms of water uptake. By the way, I have a metaphor for you. So think of a corn dog. And so the hot dog itself is the root tip, and the cornmeal covering is the mantle, the sheathing mantle around that. Right? And don't even think about it if you put that corn dog in water. It's going to be a mess, but there's going to be a lot of water uptake, right? Okay. All right. Okay, real quick, we'll review some of the mycorrhizal types. And I have some pretty pictures for you, and then I'll take questions after that. So, so here is the Ectomycorrhizae with the sheathing mantle and the Hartig net. Over here is basically the complete opposite of that, with the says VA endomycorrhizae. It's basically AM mycorrhizae, with very little going on outside of the plant. But with these arbuscules and sometimes with these vesicles inside of the plant, inside of the roots. Here's the orchid endomycorrhizae, which is very similar to the AM mycorrhizae, except it has the coils inside. And then the ericoid ectomycorrhizae is very similar, or sorry, endomycorrhizae is very similar to the orchid. And then finally the arbutoid, which is most closely similar in structure to the Ectomycorrhizae. All right, that'll help you out with the exam, by the way, this slide. Oh, I forgot to talk about Suzanne Simard. But actually, before we talk about Suzanne, let me take some more questions if there's some out there.
Yeah, sure. There are quite a few.
Great.
I'll go from oldest first. Andrea asked, can certain fertilizers kill the mycorrhizae?
Probably not fertilizers. Fertilizers, are mostly, if we go back to there. If you look at this list over here, that's what's in fertilizers, right? So ammonium and nitrates and so on. So it actually may enhance the growth, but I don't know for sure if it would do that. But it probably would be. It's unlikely that it would kill it.
Righto. And so Brian asked, what is the evolutionary relationship among the different sorts of mycorrhizal associations?
Yeah, boy, I wish I knew that. I'm not sure it's even well understood. What I can say is that the predominant theory about mycorrhizae as a whole is that they were originally completely parasitic. So if you think about that, there's a lot of parasitic fungi out there. And so if you can imagine that here's a fungus that, infects a root, and it's basically getting carbohydrates from that root, it's stealing the photosynthates, but at the same time, it's getting nutrients for its own benefit. And then at some point in the evolutionary time, the tree actually, due to passive diffusion, basically gets those nutrients, some of those nutrients, the potassium and the phosphorus and so on, from the mycorrhizae. So I, again, I'd just be guessing, but I would say that probably arbutoid is probably the most primitive of them, and actually monotropoid, which is actually more complex, kind of reversed the system later and became parasitic on the fungi. So basically flipped the coin. And Ectomycorrhizae probably are the most advanced. And I'm only basing that on complexity.
Righto. Robin asked, do annuals have fungal relationships or only perennials?
Certainly annuals do, yeah. Yeah, right. When we talk about 90%, yeah, annuals definitely have this. And for the most part, it's going to be arbuscular mycorrhizal relationship. Yeah.
And then Peter asked, I think this is in relationship to the monotropoid, why isn't it using the carbohydrate instead of stealing it? I don't know if
it uses it. So basically they're utilizing the carbohydrates, but they're not making it themselves, right? So they're stealing it from another plant via the fungi.
Right. Alice asked, how can you distinguish mycorrhizal species? Visually under a microscope or with gene sequencing?
Oh, you mean like a root tip core, like my colleague was doing when I did my thesis? I think that's what you mean.
How are you identifying them?
Right. So back then, now we're talking about in the last century, when technology was not where it is now. My colleague was using what are called RFLPs. So that means restriction fragment length polymorphisms. It basically boils down to it's a technique of DNA fingerprinting. And if you use the right, what they call primers, which are chemicals that cut, sorry, not primers, restriction enzymes. They're chemicals that cut the pieces of DNA into chunks. If you use the right ones, you can get information at the species level. If you use different ones, you could get maybe information at the family level. And if you use different ones, you could even get information at the individual level, right? So it depends upon how you work that. Now, that was back in the 1990s. These days, they're using sequencing techniques, things like that, right? So basically DNA fingerprinting.
Stardust asks, where do the psychedelic components of psychoactive mushrooms come from?
Oh, those are not mycorrhizal, so I don't care. So those are actually in a completely different category. Those are saprotrophic. And the fact that, for example, that you can grow psilocybe mushrooms in your garage is a good indication of that. If you had a mycorrhizal species, you'd have to grow that with the tree that it had the relationship, or the other plant, right? But I don't know. I am not a chemist, although my mycological father, a guy named Michael Bugh, that was his thing. He was actually looking at psilocybin, psilocin, baeocystin, and how all of those chemicals formed in the psychotropic fungi. But I'm sorry, I'm not a chemist. I can't answer your question.
Righto, understandable. Susan asked, do annual plants and seeds participate, exchange nutrients using mycorrhiza? But I think maybe you already answered that, right?
Well, yeah, but there's you're kind of dancing around how I'm going to finish up.
Oh, okay. So to look forward to. Okay. Yeah. Mary asked, I bought garden soil for my raised beds that appears sterile and the plants did not grow. Can the soil be inoculated? And along those same lines, if you don't mind me putting a similar question, are there, if you're doing relationship restorations, like native plant restorations, how long does it take for these relationships of mycorrhizal relationships to reestablish?
Yeah, so yeah, that's a real interesting sort of field of study that I think is still in its infancy. So it's basically where do these mycorrhizae come from in the first place? So here you got a seed, ends up in a substrate, it germinates. So where does the fungus come from? Was it already there? Did it find the seed, or did it find the root of the seed after a while, or was it there? So those are some really good questions, and like I say, I'm not really sure about the state of understanding of that right now, but I don't think it's very far along. I do know that you can purchase mycorrhizae in like packets, like dried mycorrhizae. I have to tell you, I'm pretty skeptical about that. I don't know if that actually works or not, but who knows? It might. As far as the soil from potting soil, I'm kind of surprised that it didn't work, because there are a lot of spores just in the environment, and you really have to work at keeping soil in a pot sterile. You'd have to kind of keep it in a greenhouse maybe, or with a reverse osmotic flow in there. You'd have to kind of make sure that that there are no contaminants. That's why people who grow mushrooms have these sterile rooms and the ventilation systems that are very complicated. So if you were just planting a plant in a pot in your backyard, chances are there's going to be some spores from fungi ending up in that pot. And so there's probably going to be mycorrhizae. So there might be something else going on with that particular situation that you've got. Even this in the production of potting soil, I don't think they sterilize it, and even if they did, it could easily become contaminated, quote unquote, contaminated with some fungi at some point. Not in a bad way. Fungi are good as far as I'm concerned, even
Right.
the ones that you don't like.
Necessary. Absolutely. So another question, how are the 6 types of mycorrhizae related to families, genus? Are they Is that a
There's
complicated?
some that are, there's some that are, and some that aren't. So the orchid mycorrhizae only found on orchids. So this may be kind of going back to somebody's question about evolution. So the characteristics that we see in orchid fungi are only or orchid mycorrhizae are only found in this relationship, not any other relationship. Arbutoid and ericoid and monotropoid are all found only in ericaceae and not anywhere else. And monotropoid are only found in this mycoheterotrophic. So these four middle ones here are very specific to their hosts. Arbuscular, you know, what would I say, 300,000 species of plants have arbuscular mycorrhizae. And then Ectomycorrhizae to a lesser extent, but still across a broad range of groups of organisms, from gymnosperms to angiosperms and, you know, everything in between, you know, within there. There's actually even some arbuscular mycorrhizae that are found with liverworts and mosses. Yeah.
Wow. So David's got an interesting question. You said 90% of plants have these mycorrhizal relationships.
Yep.
Which ones don't? What kinds don't?
So let's see. You know, a lot of them are tropical. And if you know much about tropical systems, the soil is actually very thin and there's a very high nutrient turnover in tropical soil. So a lot of those plants have evolved to be able to cope with that. And part of that is basically getting nutrients without any help for that. But one sort of the most, the poster child, at least in North America, are members of cruciferia. That kind of ages me, right? Sorry, Brassicaceae. Yeah. So the Brassicaceae is a whole family that does not have mycorrhizal relationship.
Oh, wow.
And then there's a few species here and there in other families.
Cool. Carrie works in a California native plant nursery and was doing some research, added a commercially available mix of ecto and endo because it sounded like they work in conjunction with each other. Added some Quercus, Arctostaphylos, Ceanothus containers, and all seemed to respond. What are your thoughts about using MRF in a nursery situation?
Yeah, again, I have to say that I'm skeptical about that, and I would I haven't looked, actually. I should do this before I give this talk again, but I haven't looked to see if there's actually been some well-controlled, peer-reviewed papers on the efficacy of those additives, the mycorrhizal additives. I would say that also that there's probably it probably doesn't having both probably is not a benefit. So, for example, with Manzanita or Ceanothus, you would have Ectomycorrhizae in there, and the arbuscular mycorrhizae are going to get out competed by those Ectomycorrhizae. So even if that works, I'm not sure having both of them will work. But it's kind of like if you're going to be marketing these, you might as well put them both together because it's a shotgun effect, right? So you're going to put this stuff in the soil, and again, assuming it works, maybe only one of the strains will work and others don't. But there you go. That's okay. It works, right? Whereas if you didn't have that particular strain, it wouldn't work. So I think that's sort of a marketing tool, and it actually makes it easier too. Again, assuming it works.
Right on. Susan Mulaney's got an interesting question here, but it's might be it's more about chemistry. We know that chloramine will kill aquatic invertebrates, so and it kills a lot of insects. So chloramine is what they put in drinking water. to keep us from getting sick, basically, but it's also used heavily in irrigation. And we've learned from researchers that it will like knock out a lot of your beneficial insects, you know, if you use it for like native, you know, habitat restorations, that kind of thing. So, does it also, do you know if it affects mycorrhizae?
Logic says yes, but I don't know for sure.
Yeah. Yeah, I think it's, I'm pretty sure it just works on critters, and but anyway,
fungi are more closely related to animals than they are to plants. So,
right,
you know,
yeah,
so
yeah,
depending upon the chemical mechanism, it could affect fungi in addition to animals. Yeah.
right. We there's a very curious audience out here, by the way. There's still about 6 or 7 more questions. You want to tackle them now?
Yeah, let me talk about the work that's done by this woman. Suzanne Simard, she was the talk of the town last summer, the summer before last, in 2019. I don't know if you heard about that. She was on. She did her TED talk, by the way, and there's a link there. So if you just Google Suzanne Simard TED talk, it'll come up. It's really good. You should check it out. I'm not going to show it to you, but you should check it out. And then she was on Science Friday, and she was, you know, interviewed by all kinds of people. She was the talk of the town. So what you know, her approach, or her hook is how trees talk to each other. Now, when I saw that title, I was like, I cringed as a scientist. It's like, you know, the trees don't have vocal cords, they don't have ears, you know. So you know, and maybe I would feel a little bit better if instead of talk to, replace that with communicate with, right? And even then I would kind of cringe about that. But Suzanne really did some amazing work a long time ago, actually. So I think in the 80s, or maybe the 70s or the 80s. So she's the daughter of a forester from British Columbia. So she grew up with trees. She was fascinated with them. And so when she went to college, she went to college and went into forestry, I think at UBC, and studied plants. And so she was curious about whether or not there is whether or not trees can actually communicate with each other via the mycorrhizae. And she did some very elegant work, and actually found out that yes, they do, that there is strong evidence that there are chemicals, communicative chemicals, that are passed from one individual tree to another individual tree via the mycorrhizae. Now, when she basically went to publish this, she was not recognized as being, it was not recognized as being real science, and which is really unfortunate. You know, it may have a lot to do with the fact that she's a woman, and back then in the 70s and 80s, you know, it's a men's world in the science. But she has since continued some of her work and became a celebrity last summer, belatedly, but rightly so. So here are some of the things that she came up with. So there is an exchange of carbon and other nutrients via mycorrhizae. Now, this has actually been shown many times, and this is actually how. So actually, let me back up a little bit. So it's pretty easy to do. So you can take some carbon-14 or some other isotope. Carbon-14 works really well. Put an isotope in the phloem of one plant, and then you just track it with your isotope detectors, your radioactive detectors, and you can tag it. You can see what happens, and indeed, it shows up in another plant. So some work by Tom Bruns over at UC Berkeley was done on Douglas firs and Arctostaphylos up on Mount Tam, actually, showing this was happening. So there's some exchange of nutrients between plants for sure. And then what I was about to say is that this is how the mycoheterotrophic relationships kind of was discovered as well. You know, so the here you've got this snow plant or this coral root orchid, and how is it surviving? Well, then you can track that with carbon-14 from a tree through the fungus and into the Achlorophyllus plant. Okay, and it turns out that she did some work with Douglas fir and birch up in British Columbia, and she found that not only is there exchange of nutrients between two Douglas firs, but between Douglas firs and birch trees, and backwards, back and forth, right? She found out that parent trees are more likely to do this kind of communication, do this kind of nutrient exchange with their own offspring. And there was also, this is found out later, that she found out that older trees that are in poor health, so for example, infested by beetles, sent specific communication chemicals to younger, healthier trees, which then created an immune system response, basically, from those younger trees to kind of prevent the infestation of these beetles on the younger trees. Crazy stuff, really, really exciting and very, very fun stuff. Now this illustration here, which is really cool. So this is a population of I think Douglas firs. The size of the circle represents the age class. So I think that there are oh, sorry, wait a minute. It's the color that represents the age class. So there are 4 age classes, with the yellow ones being saplings, and then the big dark green ones being the oldest trees. And then the size of the circle has to do with how many connections they found in this grove. Totally cool. Really neat stuff. All right, well, I'm going to end with some pretty mushroom pictures of a few mycorrhizal species. And then I'll be happy to stick around for questions. Okay, so let's see. This is an Amanita. This is an Amanita bisporigera from the eastern United States. This is also called the destroying angel. It's highly toxic. It has the same toxins that our death cap has out here. We actually have another one called the destroying angel, different species than this, but a white Amanita. All Amanitas are mycorrhizal, including edible ones like this one. This is Cora. This is a absolutely delicious mushroom, but you want to make sure you identify it correctly. Again, an Amanita, all Amanitas are mycorrhizal. This is Lactarius deliciosus. Not all that delicious, but it is edible. It's a Lactarius which forms this latex. Many of them have white latex or yellow latex. This one has orange latex. And all of the Lactarius and the other members of that family, Arbusculaceae, are mycorrhizal. This is Matsutake. So this is a highly sought after edible, highly prized in Japan and other parts of Asia. Mycorrhizal. This is a Cortinarius ponderosus. All of the Cortinarius, which is a huge genus, mycorrhizal. This is related to Cortinarius. This is a Phaeocolybia, really great mushroom. You can see what's showing above the duff here. And then actually you can see that below in the ground, they have these long rooting stipes that go deep, deep, deep into the soil. Actually, I broke off most of these here. Phaeocolybia. This is Croagomphis, which is closely related to this one, the Gomphidius, what David Aurora called the hideous Gomphidius. The Croagomphis and Gomphidius are both mycorrhizal. In fact, the Croagonthus' common name is pine spike, and mycorrhizal with pines. Here is a white chanterelle, absolutely delicious, and mycorrhizal. Here's a relative of the chanterelles. This is a coral mushroom. So there are some coral mushrooms that are not mycorrhizal, that are saprotrophic, that are decomposers, but many of the coral fungi are mycorrhizal. This is a related species which has a huge presence on a lot of plants, and this is called a Thelephora. You don't see it very often. It really blends into the duff, as you can imagine, but it's very, very well represented on Arctostaphylos and on pines and Doug firs and lots of different species. This, by the way, is the only photograph that's not mine in this little sequence. This is actually Boletus edulis from Europe. This photograph was taken last winter by a friend of mine in Germany. And almost all of the members of the Boletaceae family are mycorrhizal, including our Boletus. This one right here is Boletus edulis, variety grandif something. Grandis, I can't remember. Anyway, it's the porcini that grows here in California. And then a lot of the underground mushrooms grow here. This is one that we found in a tree, probably put there by a flying squirrel or something like that that had dug it up and placed it there for later use. They don't grow on trees like that, but they grow underground. So basically the false truffles and the true truffles. This looks like it's probably a rhizopogon or something like that. And then a slideshow is not complete with a picture of my granddaughter and a mushroom, which is not mycorrhizal, but that's okay. It's my granddaughter. All right, thank you very much.
Wow, that was really fascinating. Lots of, I mean, it's mind-boggling too. Some stuff, especially the relationships that you were describing of the trees. We saw that talk some time ago now, and it was. I'm glad you brought that up. There are 11 questions in the Q&A. Are you and a few more in the chat actually? Are you
okay. Yeah.
Are you up for it?
Yeah, I'm up for it for sure. Yeah, so if you have to leave, it's 9 o'clock already, so bedtime for some of us. But if you have to leave, thanks for coming. But yeah, I'm perfectly willing to stick around as long as it takes, as long as there's questions. Happy to do so.
Cool. Yeah, and would you want to say anything about the Sierra Nevada Field Campus?
Oh yeah. Let me bring that up. So, earlier on, you asked if it was going to happen, Eddie. I'm not so sure it's going to. That was a fingers crossed, actually, not a thumbs up.
Oh, okay.
Yeah. So it's kind of a situation. We didn't have a program this last summer, clearly. And it's a situation where it would be very difficult to make it safe for people to have classes. It's difficult to distance and so on. But who knows? Who knows? A vaccine could come around the corner, and mass production and ways to deal with that. So we are putting together a program for next summer. But certainly in 2022 there will be one. Absolutely. So as Eddie mentioned, we have a lot of natural history courses. So plants, obviously, we have a great mushroom class that is held up there. Several bird classes, bats, astronomy, lots of things like that. A lot of art classes, so watercolors and pastels, and mixed medium classes. We have a writing, writing class. We have sometimes we have fly fishing classes and wilderness first responder classes and so on. It's really great. We can feed about 50 people in our dining hall, and we provide 3 meals a day: hot breakfast, hot dinner, sack lunch, and we have tents that we provide for our guests at a premium, I have to say. We have 18 tents, and then a lot of people bring their own tents or stay off campus. So it's a really great program, and you know, I bet there's a bunch of you out there who have had the good you know, pleasure to be there. You included, Eddie, and checking it out. And
real time.
anyway, yeah, thanks for letting me do the plug.
Absolutely, yeah. No, I want more and more people to go there, but not so many that we can't come.
Yeah. Oh, don't worry. There's always room for Golden Gate Audubon.
Yeah, so there's I don't know if you want to take these questions, just open your Q&A and
That's a good idea, actually. Yeah. So let me see if I can figure out how to do that. Do I have to stop sharing?
I don't think so. You could probably just go down to, you should be able to hover down to the bottom and pick up Q&A.
Yeah. All right. Okay, so let's see. Some mycorrhizal types form with plants that are evolutionarily so far apart. Yeah, that's really interesting, isn't it? So I think, you know, I think it's kind of like even let's see, what's a good example? Oh, like pollinators, for example. So you have some pollinators that are specialists, right? So they only pollinate one species of plant. And then you have other pollinators like honeybees, for example, that are generalists. And so it's the same thing with fungi, with regards to mycorrhizae and with regards to saprotrophic things. So some will only decompose, you only find them on pine cones. Other ones you find only on oak leaves, right? And then other species you find growing anywhere. So I think it just has to do with that there's so many different kinds of fungi that there's going to be specialists and there's going to be generalists. And I think that's what my colleague was finding with the root tips as well. So and actually with my work, so some of the mushrooms I found under Arctostaphylos were also growing with pine trees. Completely different parts of the plant kingdom, but still able to take advantage of that, of both types. Okay. So Rhonda's question about transplanting native plants, you know, again, I think you just have to trust the dirt. So the spores are going to be there. There's going to be an opportunity for them to take advantage. Now, how that exactly works, I'm not really sure. I don't think anybody knows for sure, but certainly just kind of you just have to really trust it. Now, sometimes what will happen is you'll get a little lag time. So you have to take care of that plant. You know, you have to water it, maybe fertilize it, but eventually it's gonna find the right mycorrhizal fungi, as long as it's not sequestered from that, like in a greenhouse or something like that. Why do hyphae kill springtails for nitrogen? Yeah, there's some predatory fungi out there going for springtails or nematodes and so on like that. So that's basically, they're just taking advantage of that. It's a source. I teach an ecology class, and we talk about the niches, right? So springtails and nematodes and other small critters like that are basically opportunity for the fungi to get a hold of some nitrogen through their protein. Right? Do invasive plants affect mycorrhizae in the soil? That's another one that's really not well understood. Probably. There's probably a oops, I think I'm losing my track here. Oh, I see. It went into the answered questions. Anyway, yeah, probably there's some effect. So, for example, eucalyptus are mycorrhizal themselves, and they have a suite of fungi that grow with them. And if you go to Australia, it's one of the things you want to do is go mushroom hunting in the eucalyptus forests. and you find completely different things there. So I'm sure that they're kind of displacing some, and others are actually taking advantage of that, and so on. And it can kind of go the other way too, right? So for example, the most deadly mushroom in the world, the death cap, Amanita phalloides, is an introduced species. It's not native to North America. Okay, Jake's question about radish or mustard. You know, I don't have no idea, Jake. Again, my gut feeling would say, so Jake's question was about inoculating. Do you need to inoculate mycorrhizae into the soil after you've had, you know, a wild radish or mustard in a plot? I think, you know, again, my gut feeling is that you just trust the soil, trust the dirt, that there's going to be seeds, spores and hyphae of mycorrhizae species that will take over. Now, there may be other things going on. So, for example, you know, like ice plant, right? So, ice plant actually produces allelopathic compounds that prevent anything else from growing there right after you've pulled it, and you have to you have to amend the soil or wait till it recovers. So maybe that may be the case with the brassicades, but I don't know for sure. Okay, Abdul Aziz asked if there's a relationship between root vegetables and carrots and mycorrhizae. Well, carrots are in Apiaceae, right? The parsley family or the carrot family, and those are all, I think, I'm pretty sure they're all arbuscular mycorrhizae. So there's probably there. So when you're growing root vegetables, they're probably utilizing the mycorrhizae that's in the soil that they're growing in, right? But it's AM, so it's not going to produce mushrooms. The recent fires on the soil, yeah, that's really interesting stuff, huh? And there's a lot of work, and I have to tell you, I'm not fully read up on that. That'd be a completely different talk, actually. But Tom Bruns over at UC Berkeley, some people call him Tom Burns because he has done a lot of work on this, and he had some pre-fire data from the Inverness Ridge fire back in the day, which was Point Reyes, and swept through the Bishop Pines there. So he had some data, some soil cores and things like that from the Bishop Pines in the area that then later on got burned. And then he's also done a lot of work. He and his lab have done work on the Ridge. Is it the Ridge fire that was up by Yosemite? No, the Rim fire, which was really an interesting fire because it burned super hot. And so I know a little bit about that one. So some of the fungi were just wiped out because of the heat. But if the fires move through pretty fast, it turns out that the mycorrhizae do okay. So again, that's a whole study that's really fascinating. Okay, Perry has a question. How can you tell the difference between mycorrhizae and the bad type of fungus that grows, for example, on a tree trunk? Yeah. So basically, okay. So first of all, remember that most mycorrhizae don't make mushrooms. So they're microscopic. They're on the roots. You don't even know they're there. So I assume that what you're talking about are the mycorrhizal, ectomycorrhizal fungi that I showed at the end of the talk here. So amanitas and chanterelles and boletes and things like that. Now, it's basically a matter of identification, and there are not that many mushrooms that are actually bad for your trees. And the major culprit there is a genus called Armillaria, or honey mushrooms is the common name for Armillaria. And if you have one of those growing like on one of your trees, that's bad news because it's parasitic. Now, don't confuse, and by the way, it's just again a matter of just learning how to identify it, how to recognize it. using a field guide or going to Mycological Society field trips when those start happening again or whatever. Now, don't confuse that with shelf fungi that are growing like on a living tree. Those are actually saprotrophic. They're not parasitic, and they're eating the wood, which is dead, right? So if you see a shelf fungus growing out of a tree, it doesn't mean that that tree is being parasitized. However, it can be bad in the sense that it weakens the tree, although typically it's sort of the other way around. Once you see those mushrooms, it kind of means that that tree is sick and dying, and that the mushroom's now taking advantage of that. But again, those are not mycorrhizal at all. If it's growing right out of a tree, not mycorrhizal. Yeah. All right, Joyce has a question. What conditions encourage mycorrhizae in the soil? So you do habitat restoration, you have bare soil on slopes. Again, trust the soil. I'm really skeptical that anything such as the mycorrhizae that you purchase in the hardware store really works. I mean, it might, but again, I haven't seen good evidence of that myself. I haven't looked for it, but I haven't. But anyway, I think a lot of the bare slope issue, the issue with bare slope is compaction, right? So when you don't have anything growing there, the soil gets compacted from people walking on it, or animals walking on it, or just from drying out and losing the spaces between the soils. And when that happens, then you lose a lot of things, not just mycorrhizae. You lose the soil engineers like earthworms and termites and ants and things like that. You lose the ability for plants to colonize that, to come in and recruit there. So and I have to say, I'm not a gardener, and I'm certainly not an expert on habitat restoration, but that's my gut feeling from your question. Just trust the soil, trust the dirt. Anonymous, is it true that the exchange of carbohydrates and nutrients between a mycorrhizae and a fungus and a plant can be subject to negotiation or even reward punishment schemes? So yeah, I don't know. Boy, I wouldn't be surprised, considering Suzanne's work on communication between different species of plants. There very well could be some sort of a communication system or sort of action response between a plant and a fungus, where if the plant doesn't provide enough, then the fungus responds accordingly. I'm not sure, but I wouldn't be surprised if something like that does happen. Yeah, there you go. There's a PhD for you. All right. Longen asked a question: does one species of plant associate with only one or more than one species of mycorrhizae? Yeah. So it can be both ways. So, for example, with Suzanne's work, so she found that the birch trees and the Douglas fir trees were sharing mycorrhizal networks. And so, yeah, so that certainly is a circumstance. And again, I don't think there's a whole lot known about that. It's an elegant. So what she did was elegant in terms of trying to figure that out, but it wasn't easy. And if you listen to her TED Talk, you can find out more about the trials and tribulations of her work, and grizzly bears, yeah. Anyway, and Vivian asked, last spring I had morels come up out of a wood chip mulch at the backyard. Morels are morels mycorrhizal, and can you encourage them to come back? They're not mycorrhizal, as far as we know. And in fact, there's some apparently some success in cultivating them. which is very difficult with mycorrhizal species. The only thing I know of that's been cultivated for as far as mycorrhizal species are certain truffle species, and you have to have the tree. So morels are not mycorrhizal. You are probably really lucky to have morels growing in your own very own wood chip pile. I hope you enjoyed them, but don't hold your breath. May or may not happen ever again. So,
Wow.
and are there some in the chat, Eddie?
Yeah, but I think you pretty much covered the subjects I saw,
Okay.
and there was a lots of thank yous and gratitude for you.
Great.
And I also give you much thanks and gratitude for a really wonderful, wonderful show.
All right, my pleasure.
Yeah, thanks again, and I hope we get to see you in person sometime.
Yeah, I'm sure it'll happen, and you know, I have a couple of other talks, so maybe maybe I'll see CNPS real people face to face live someday.
Yeah,
I look forward to that.
sure hope so. Thanks again, JR.
All right, take care, everybody.
Everybody for joining us.
Thank you.